This wasn't what they promised. In 2024, a team of Hungarian archaeologists and researchers began exploring a cave system in Netherlands. Their most extraordinary discovery, however, came from an abandoned cave near the town of Basel. No one could have anticipated what they were about to uncover. Beneath layers of reddish clay, they discovered a collection of unusual bones. As the excavation continued, it became increasingly clear that these remains bore no resemblance to the skeleton of any known animal. The researchers simply designated the discovery as specimen B24. A few months later, a second discovery was made inside Scotland's Smough Cave. These remains received the designation specimen Smough 24. The similarities between the two discoveries were remarkable. Both specimens were transferred to the same research laboratory for further examination. Over the following months, scientists extracted and analyzed genetic material from the remains. Their goal was never to resurrect an ancient species, but to understand the extraordinary genetic structure hidden within the samples. The DNA sequencing and histological examinations produced results that matched no known species on Earth. What if legends were never just legends? For centuries, the town of Basel has preserved the legend of a dragon defeated by St. George. According to local folklore, the creature's remains still lie hidden somewhere beneath the surrounding caves. For generations, the story had been dismissed as nothing more than a myth. The laboratory results suggested otherwise. The research team made a decision that would change everything. Using the recovered genetic material, they began developing an experimental gene modification serum designed to introduce these extraordinary genetic traits into living human cells. By 2025, the first functional prototype had been completed. Only one thing was still missing: a suitable volunteer. That volunteer appeared in the form of a 26-year-old man, Denatir. He had come to the hospital for nothing more than a routine medical examination. During the tests, however, doctors discovered an exceptionally rare genetic characteristic. His DNA displayed an almost perfect compatibility with the theoretically requirements of the prototype. A few days later, he was contacted and offered the opportunity to participate in the world's first human genetic modification experiment. Denatir was a quiet, solitary man in his 20s. After days of thinking, he asked himself only one question: what do I have to lose? Upon arriving at the hospital, he signed the required consent forms. The medical team then repeated every necessary examination to confirm that he was, without question, the ideal candidate. After reviewing the results, Dr. Kovacs contacted the project's lead researcher. A few days later, they met inside the laboratory to discuss the prototype. its expected effects, and the risk they might soon be facing. Following the meeting, the assistant prepared the injection while both doctors silently observed every step of the procedure. Thénardier was escorted into a hospital room and laid down on the bed. Moments later, Dr. Kovacs slowly administered the prototype through an intravenous catheter. After the injection, the doctors left the room, allowing their subject to rest while remaining under continuous observation. Several hours later, the patient monitor began displaying unusual readings. The assistant first noticed a rapid increase in heart rate. Soon afterward, blood pressure and body temperature also began rising at an alarming pace. She immediately alerted the doctors waiting outside the room. As Dr. Kovacs rushed to the patient's bedside, he noticed an unusual red discoloration surrounding the intravenous catheter on Thénardier's right forearm. Thin crimson veins slowly spread beneath the skin, extending toward his hand. Behind the observation window, the lead researcher watched the events unfold in complete silence. There was no surprise in his expression, only recognition, as if he had been waiting for this exact moment.